Gregor Mendel was an Austrian monk working in what is today the Czech Republic in the mid-1800s on pea plants. Laid the entire foundation for modern genetics with just simple tools. He had a garden full of pea plants, notebook, and a pen. That was it. Now, he didn't set out to make this grand discovery, but he did. You may not realize it, but when you go to buy a packet of seeds, whether that's peas or another plant, you get a predictable result. And it was Mendel's work that laid the groundwork for that. You could buy peas, I suppose you could buy ones that specify purple or white flowers. It's possible. More often it's determined whether they're sweet versus starchy, yellow or green peas, the dwarf variety or beans like bush beans versus vines. All of those are traits that Mendel looked at and established. How can we breed these and get predictable results? Gregor Mendel was a gardener like me. Actually, here are my pea plants growing right now, getting a bit of a slow start here in May. But he actually had a background in agriculture. He knew what he was doing, and he selected peas knowing that the choice of his research organism would be important, partly because of the flowers and the way peas work. If he was going to control breeding of these plants and trying to cross one plant with another to see what their offspring would look like, he needed to be able to control that. And unlike most flowers who have the pollen producing parts, the anthers, out in the open along with the female parts that will be pollinated, peas close theirs in. So here's the front view of the pea, and here's the side view of the pea flower. And this central part right here is called the keel. It's closed, and that hides all the reproductive parts inside. That was part of the whole magic to his experiment. But the other part was knowing he had to choose an organism with some pretty distinctive characteristics, where there was distinct differences. So for flower color, there's purple and there's white. And there wasn't an in-between, and there wasn't 18 different flower colors. There's just 2. And he looked at 7 different characteristics that had just 2 traits. And so here they are, and you can see there's all kinds of different stuff that he looked at, how tall and short they were, whether the flowers were at the top or at the base of the leaves, all kinds of different stuff. But this was the key that allowed him to really understand what was going on. And he did a lot of work. He was no slouch. He worked for 7 years on something like 28,000 pea plants that he recorded in his notebook. Wrote down what the parents looked like, what the offspring looked like, and then basically ran the numbers to see what he could figure out. Now, one of the first tasks in this whole experiment that he did was to establish true breeding plants. True breeding means you know that every time that you get seed from that plant, it's going to be the same. And so what he did was made sure that purple flowers would only breed with purple flowers, and that white flowers would only breed with white flowers. You collect the seed, you plant them, you get white flowers. You do this for a few generations, and you're sure that these are basically pure lines of genetic material, that you only have one gene or allele in there for flower color. That was an important starting point for him. Otherwise, it'd be a mixed bag. He'd say, I have this flower and I don't know what genes or alleles it has, and same over here, it's a mixed bag. But he had to establish that first. For the next part, after he had those established, he wanted to crossbreed different types of plants. And to look at what he did there, we need to look inside this flower. Well, this is where Mendel's knowledge of agriculture and specifically flower structure came in really handy. He knew that there were these yellow, the male parts, and in the red here, the female parts of the plant. So this kind of looks like a mini pea pod, right? These are the ovules. They're not yet pollinated, but they will become the peas. So this is actually kind of a nice little setup here, right? He's got this whole package of the next generation. It's kind of like a whole litter of puppies, but this thing happens a lot quicker than raising puppies. Actually, he originally wanted to do breeding experiments on mice, but the abbot didn't want that, and it would have taken a lot longer as well. So before this flower opens, these male parts can spread pollen onto the female parts, and that's usually what happens in pea plants, because remember, those are closed. That keel is like two praying hands, appropriate I suppose for a monk, where all this is closed inside, and so the male parts pollinate the female parts. They get pollen on them. What he did to control breeding was, before this flower was fully mature and the pollen was ready to be spread, he would come in and open up the flower and remove the male parts. Now this flower wouldn't pollinate itself, and he could take pollen from another plant and dab it onto the stigma, the part where the pollen usually sticks on the female flower, and control what genes were spread to that flower. Now he's successfully created a cross between these two, and you often hear this term, you cross two plants. It just means you breed them together, and this was his way of controlling it. And then he'd write it down in his notebook. Parent A had this, parent B had this, and then you wait and see what happens. You got to wait a little bit longer. It took a lot of patience. He didn't just look at these peas and say, oh, that's the results. He had to plant each of these in the soil, wait for them to grow up, see what they look like, record their flower color, position, height of the plant, color of their peas, etc. So this was a lot of careful record keeping. Some really top-notch science going on here. All right, so here's the experiment. We have these two true breeding lines, right? So they're true breeding, which means genetically they're essentially pure. This one is only going to pass on white alleles, and this one will only pass on purple alleles. And so this is the parent generation. And so when he crossed these, remember he opened them up and he made sure he put the pollen from the white flower onto the purple flower, probably did the reverse as well, got peas out of that, planted them, and then the next generation, the first filial generation or F1 generation, this is what happened. All the flowers were purple. Planted the peas, and every single one that came up, the next generation was purple. Now this was counterintuitive. At the time, most people figured that breeding essentially was blending, right? They actually thought that you kind of purified blood somehow and passed that on to the next generation. You hear about that in bloodlines. Then plants did something similar. They purified their juices, passed them on to the next generation. And this one should be kind of lavender, right? It should be kind of a lighter purple than this, but more purple than white. But he said, no, they're just as purple as that generation. And that's where he came up with the first of what we now call his laws, kind of the rules about how this works. And that's the law of dominance. The law of dominance is essentially, I have two traits. One of them is going to cover over the other one. And so purple is dominant to white. It's as simple as that. Now, not all traits follow this, but the ones that he looked at did. And that was part of the careful selection of peas as a research subject. And sort of our convention for sorting that out would be to say that the capital letters are the dominant alleles and lowercase letters are recessive. And so that would be purple flowers, and the lowercase would be the white flowers. Doesn't matter which letters you use, I just do it that way to keep them straight. Otherwise, why does P stand for purple and white? But this is our legend. Law of dominance, basically, first generation was all purple, the white one was hidden. But we'll see in the next generation, it comes up again. So unlike this first generation that turned out all purple, the generation after that turned out purple and white. So here's what happened. He took these plants and allowed them to cross-pollinate themselves. He knew that these were a mix of both the parents' genetic heritage. When these did that, then all the little peas grew up inside. He planted those and found that he got purple flowers and white flowers again, but always in this ratio of 3 purple flowers for 1 white flower. And this was consistent. It wasn't just like he got 4 plants and said, oh, 3 to 1, that's it. He did hundreds of these. For pea color, another trait that he looked at, he counted up 4,000 different peas and found, again, 3 to 1 ratio. The more he got, the closer he got to that perfect 3 to 1 ratio. Now you can't look at that and see it again and again and again and say there's nothing there. Definitely a pattern. And this is where he came up with the next law, the law of segregation. Now, the law of segregation is simple, but don't underestimate it. I mean, this is the foundation for most of the rest of the genetics we'll talk about. And that is that each individual has 2 traits and they pass on 1. Again, 2 traits, you pass on 1. And so, looking at this pattern, not only did he come up with the law of dominance, that some traits are dominant over others, which by the way doesn't mean better, it just means that they hide the other one, but also the law of segregation, which is 2 particles. So he figured that the purple flowered parents that were true breeding had 2 purple factors, or what we'll call genes, and that the white flower plants had 2 white genes, right? And we know that those are called alleles, the different versions, purple versus white. So 2 white alleles, 2 purple alleles, you pass on 1, and so this individual gets a white allele from this parent and a purple from this, and since purple is dominant over white, you end up with all purple plants. But the white is still there, and that's kind of how we explain why it showed up in the next generation. But when you cross these, again, you're crossing purple and white with another purple and white, so you could get either 2 purple alleles, in which case it turned out purple, or a purple and a white. Both cases turns out purple flowered. But sometimes you get a white allele from each parent and you get the white flower again. That's the law of segregation. In the next video we'll work out how you sort these out using the Punnett square and all that sort of stuff. But this is just running through his experiments and essentially what he came up with. Now, if Mendel had only looked at flower color, he wouldn't have figured out the next level, which is the law of independent assortment, the next rule. And that's because he looked at 7 different traits, and he found that each of them were inherited independent of the other ones, thus independent assortment. When all those traits are sorting themselves out in the next generation, getting passed on, they're all independent of one another. So another trait he looked at was plant height. Some are tall, some are dwarf. If you've grown beans, you know about the bush bean variety versus vines. Same sort of thing here. You found that these also come in a ratio of 3 to 1. So in that F2 generation, that 3rd generation down, breeding them the same way, 3 to 1. In the 2nd generation, it was all tall plants, but that 3rd generation, 3 to 1 every time, which reinforced the law of segregation. But the fact was that tallness didn't correspond with purple flowers. So just because you had purple flowers didn't mean it was tall, and just because it was tall didn't mean you had purple flowers. You could have tall ones with purple flowers or tall ones with white flowers. Short ones with purple flowers, short ones with white flowers. Any of those combinations was possible. This idea is at the heart of genetic diversity, that each of us are a combination of our parents, right? But we're a unique combination of traits. We're not brand new. right? I have all the same genes that both of my parents do, but in a different combination, making me an entirely different person. And when you add up more than just 2 traits, but thousands of traits, you end up with a very unique combination. And that's an important thing. It's an important type of variation for natural selection to be able to act on. So Mendel put all this together and in 1865 presented it to an audience of other scientists, and he published it in 1866. To put that in perspective, we were just finishing up the American Civil War. Washington was not even a state yet. There was only, I think, 34 or 36 states at the time. 1865. He laid the foundation for all modern genetics. The idea that every organism has 2 traits and passes on 1. Now, I don't think he really meant to. He actually went on to do other experiments on different plants and found that they operated differently. So I'm not sure that he meant to say this was the way that all genetics works. But as it turned out, it was. He kind of moved on to other things. His whole goal seemed to have been to lay out how to breed peas better. He had a very humble goal. Then he moved on to be abbot and do monk type things. He was not ingrained in the scientific community. He was actually kind of separated over there in Austria in a way, which may be part of the reason that his work wasn't really rediscovered until about 1900, after he actually already died. But there's a lot to admire in his work. It's some very carefully done science. I mean, he really took his time. 7 years worth of work, meticulous record keeping, really good analysis. He shared it with other people and was really humble about the results. He could have gone out and promoted it and tried to seek fame and fortune in the scientific community, but he just said, here's my work, take a look. I hope you appreciate it. And I do. I hope you do too.